All right, this portion of the Lab 2 lecture is referring to activities that will only be occurring for the students taking the course in person, face-to-face. For those of you who are taking it online, you don't necessarily have to watch this half of the video. However, if you have purchased the lab manual, this will help you understand what's going on or would have gone on if you had gotten to take the course in person. Anyway, so in the lab, we will be using the enzyme peroxidase to break down hydrogen peroxide. It turns out the cells of our body during regular metabolism produce hydrogen peroxide. Hydrogen peroxide is toxic to our body, so we have these special organelles called peroxisomes. The peroxisomes have many enzymes, including peroxidase within it. So the hydrogen peroxide gets sent to the peroxisomes, and then the peroxidase will break down the hydrogen peroxide into safer compounds, water and oxygen. So in the lab, we'll be using turnip juice, but if we had used liver, we also could have used liver, but I don't know, turnip juice is a little less messy in my mind. So the way it'll work is we will have hydrogen peroxide coming from a bottle. This will be a liquid. We will then mix it with peroxidase. Peroxidase is coming from the turnip juice. Peroxidase in the turnip juice. So this is the substrates. Here's the catalyst. And then we will get products catalyzed by peroxidase, where hydrogen peroxide gets broken into water, which is a liquid, and oxygen gas. So again, the products are what the arrow is pointing to. The arrow is pointing to this, so this is the product. So hydrogen peroxide with peroxidase gets broken down into water and oxygen gas. We can then collect the oxygen gas in a burette, measure how much oxygen gas is produced, and use this to determine the relative reaction rate of that reaction. Here is what the apparatus setup would look like. So there is a ring stand with a burette clamp, which will connect to a burette, which is a long tube with measurements on it and a little valve that lets air in and out. Then we have a water trough filled with water. We have the flask. In the flask is where the chemical reaction will occur. This is where the hydrogen peroxide and the juice with the peroxidase will be. And this tubing will connect to the burette. So there'll be water in the burette, water in the water trough, and as our chemical reaction occurs, the oxygen gas will move through the tubing up to the top of the burette, causing the level of water to go down. We can measure the difference from where it started to where it ended over a certain period of time, and that will tell us how much oxygen by volume was produced. So, in the lab activities, we will compare 3 different pHs. Acidic, pH of 2, neutral, pH of 7, and basic, pH of 11. We will compare what the water level is before the chemical reaction started to the water level after 30 seconds of allowing the collection of oxygen as peroxidase breaks down hydrogen peroxide. And that'll give us the amount of water displaced, and that'll let us know the relative reaction rates between these 3 chemical reactions. So again, one of them will be at pH 2, one of them at pH 7, one at pH 11. The more water displaced, the closer to the ideal pH that the enzyme is at. So that's what that part of activity would do. Then we have a similar activity measuring the addition of more juice. What will occur when we add? Oh, I skipped an activity. My bad. Activity 1. Activity 1 has to do with temperatures. Room temperature, ice cold, and boiled. So we'll have 3 different samples where the peroxidase is trying to break down hydrogen peroxide. One will occur at room temperature, one on ice, and one after it's been boiled. And we'll again compare the level of water before and after the chemical reaction is allowed to occur, 30 seconds. And the displaced amount of water gives us the relative reaction rates. And the one that has the most water displaced is the one that has closest to the ideal temperature for the turnip peroxidase. Turns out, though, I'll just go ahead and tell you part of it, that boiling enzymes destroys them. So those will be denatured. So we expect no water to be displaced from the boiled turnip juice. So then we just try to determine whether room temperature or ice cold is better for turnips when they produce peroxidase and have it functioning. pH, again, here we're comparing 3 different pHs. And then the third set of reactions, a little more complicated. What we'll be doing here is doing the standard reaction, the turnip juice plus hydrogen peroxide, measuring it for 3 minutes instead of 30 seconds. Oh, sorry, for 2 minutes, 120 seconds, 2 minutes, and seeing how much water is displaced. So, start off, put the enzyme juice with the hydrogen peroxide, see how much water is displaced after 2 minutes, and then remove the stopper and swirl and let the reaction go to completion so that all of the substrate is used up, all of it gets converted to water and oxygen. Then, in the exact same beaker, we're not doing separate reactions, this is in the exact same beaker, we will add more enzyme. We will add 2 more grams of enzyme, and we'll see how much water is displaced after 2 minutes. And then, after that has occurred, we will then add more substrate. So in the exact same beaker, where we already have 4 grams of turnip juice, we will add an additional 3 mils of hydrogen peroxide, and we will compare water level at the beginning of the reaction to after 2 minutes to see how much water is displaced. So, round 3 is adding more substrate, more hydrogen peroxide to the same flask to see what happens. So we'll wonder which of these will be the fastest, will have the most water displaced. Which of these features of the enzyme is being studied in this experiment? Well, save that answers to those questions to when you actually do the experiment in the lab. But to be honest, you should kind of be able to figure some of it out. Now, if you remember the various special features of what makes enzymes work faster or slower. All right, so the effects of various characteristics on enzyme activity, how fast the enzyme works. We will be wanting to understand temperature, its effects, what is the ideal temperature, pH, its effects, what's the ideal pH, concentration of substrate, how much substrate is there, does that speed up the chemical reaction, and concentration of the enzyme. Will having more enzymes speed up the reaction rate? And this, of course, depends on whether or not there's plenty of substrate present.